Good morning to all of you students of BT First Sem. Today I am going to discuss with you about the important topic that is given in your cell biology, that is your ribosomes. Now let's see if I can able to complete the whole part of ribosomes, because it has been given the structure of ribosomes and as well as the function of ribosomes. Um, Involving the role of ribosomes in your protein synthesis. So let's get started now. So we were doing about the ribosomes today. Now ribosomes are the sites for your protein synthesis. Generally, your if I say about the structure of ribosomes, ribosomes are spheroid in structure and its diameter is about 150 to 250 angstrom in diameter. So the diameter of ribosomes is 150 to 250 angstrom, and each ribosome is actually porous. That means it has pores. Generally, ribosomes have three sites. Ribosomes are made up of two subunits. One is called the large ribosomal subunit. I'm writing as here in short form as LRS, and the small ribosomal subunit called as SRS. Well, I am giving here because there is very short space to write. So I am representing large ribosomal subunit as LRS and small ribosomal subunit as SRS. Now all of these subunits of ribosome have three sides. There are three sides of ribosome. That's why I've said they are porous. So three sites are present in the ribosome. One is called the A site, one is called the P site, and another one is called the E site. Well, the A site is called the amino acylation site because here at this site the charged tRNA or the tRNA, which is carrying your specific amino acid, will come and bind into this site by observing the specific codon sequence in the mRNA. So if this is the mRNA and this is there is some sequence in the mRNA which is um, quite matching with the anticodon sequence found in the tRNA, then it will bind, okay, into the A site. Okay, so that's the specification of the A site. So that's why I have said that ribosome have are porous in structure. Okay. Well, what about the P site? Well, in the P site, there the main peptidyl transferase reaction is carried out, where the tRNA, which will be located in the P site, will transfer its amino acid to the tRNA in the A site. Again, I repeat, the tRNA, which will be found on the P site within the ribosome, as the ribosome is always moving from 5' to 3' direction, and the tRNA, which is carrying the amino acid in the P site, will transfer that amino acid to the tRNA in the A site, and that is done in the P site of the ribosome. Now, within the P site, there is a specific enzyme, and the name of the enzyme is peptidyl transferase. Peptidyl. Okay, I am writing in other place. Okay, so there is an enzyme within the P site, and the name of the enzyme is peptidyl transferase. So, in that peptidyl transferase, um, so what happened during peptidyl transferase? So during that peptidyl transferase, this enzyme will actually catalyze the formation of a peptide bond between the two amino acid, and to accomplish this job, the amino acid which is located on the P side, that is the tRNA which is carrying the amino acid in the P side, they are that amino acid will be removed from its tRNA, and then it will that amino acid will be transferred to the free amino terminus group of the second amino acid located on the A side. Again I repeat, so both on the P side and A side supposed to charge tRNA, that is tRNA carrying amino acid are placed. Now, during the peptidyl transferase reaction, which is also called as the elongation step of translation of protein formation, during that time, the amino acid which is attached with the tRNA in the P side, that amino acid will be removed, and that amino acid will then be attached to the amino terminal end of the second amino acid located on the A side. Okay, that means the tRNA carrying that tRNA which is located in the A side, they are already our amino acid is being attached. So from the P side, that amino acid will be removed from the tRNA, making the tRNA uncharged, and that amino acid will bind to the tRNA in the A side and catalyze the formation of the peptide bond. And so on top of the second, the first amino acid gets bind. In this way, peptide bond formation occur, and this process is catalyzed by your peptidyl transferase enzyme, which is one of the important subunits, which is one of the important component of your large ribosomal subunit. Okay, what about the E side? The E side is called the exit side. And this side is used for exiting, for removing out the uncharged tRNA from the ribosome. So uh, that's why I have said that ribosome is actually having a hollow structure, having three important sites for the purpose of protein synthesis, for allowing the charged tRNA to bind in the A site. Then at the P site, with the help of peptidyl transferase center, the amino acid gets attached to the amino acid tRNA in the A site, and then in the E side, the uncharged tRNA will be removed out from the ribosome to catalyze the next round of peptide bond synthesis. So we were doing about the structure of ribosome, and well, there are two ribosomal subunit, as I have said, the large ribosomal subunit and the small ribosomal subunit. So the large ribosomal subunit I am writing as LRS, and the small ribosomal subunit I am representing here as SRS. Well, well, in prokaryotes and eukaryotes, their ribosomal subunits are a bit different, but all of them have the same thing, one large and one small ribosomal subunit together forms the ribosome. Okay, now here we found two types of ribosome here. One is the prokaryotic ribosome, another one is the eukaryotic ribosome. Well, in the prokaryotic ribosome, that is your 70S ribosome, prokaryotic ribosome is called as 70S ribosome. Okay, and this 70S ribosome is divided into large and small ribosomal subunit. The large ribosomal subunit is called 50S, and the small ribosomal subunit is called 30S. Well, we know that 50 plus 30 equals to 80, but we write the total ribosome as found in the prokaryotes as 70s. Why? Because it's due this actually we measure this ribosomal subunit in this way according to the sedimentational coefficient that we observed during density gradient centrifugation. The S that you are seeing here, it's actually it is a unit called Swedberg unit. Okay, Swedberg unit. Well, the Swedberg unit that I am trying to say with you, that in simple words, Swedberg unit is used to measure the particle size, here we are measuring the size of the ribosomal subunits, the large and small ribosomal subunits, indirectly based on its sedimentation rate under acceleration. That is, how fast a particle of a given size and shape settles to the bottom of the solution. Again, I repeat, Swedberg unit often refers to the measurement of a particle size indirectly based on the sedimentation rate. So at what rate your the ribosomes here, the ribosomal subunits here settle to the centrifugation tube under acceleration, that is how fast a particle of a given size and shape settles to the bottom of a solution. So that is called Swedberg. That's why we write the Swedberg unit when we want to calculate that, about to know about the size of the particle. Here we are trying to know about the size of the ribosomes. Okay. So I hope you have understood what I have said with you about this, about the Swedberg unit. So what I was talking about, that about the prokaryotic ribosome. Prokaryotic ribosomes is made up of large and small ribosomal subunit. So if this is the large ribosomal subunit mentioned as 50S, and the small ribosomal subunit mentioned as 30S. Together we call them as 70S ribosomal subunit. The large ribosomal subunit in prokaryotes, that is the 50S, is made up of 23S 23S rRNA. I am writing here, okay? So it will be a little, so it is made up of 23S rRNA, that is small ribosomal RNA, including it is also made up of 5S rRNA and about 34 different types of other ribosomal proteins. Okay. About the small ribosomal subunit, the small ribosomal subunit of prokaryotes is made up of 16S rRNA. Okay. The main component of your small ribosomal subunit is your 16S rRNA. Okay, and including 16S rRNA, it also have consist of about 21 different types of ribosomal proteins. Okay, so this is the whole you can say composition of your large and small ribosomal subunit found within the ribosome in prokaryotes. Well, as I have mentioned about the 23S rRNA, that so in the within the 23S rRNA, it is the main site for the peptidyl transferase, which are involved in peptide bond formation between the tRNA in the P site and tRNA in the A site. So within the 23S rRNA, your peptidyl transferase center is present actually. Peptidyl transferase enzyme is present actually. And 16S rRNA is very very important, because with the help of this 16S with the help of this 16S rRNA with the help of this 16S rRNA, it will this small ribosomal subunit can grab onto the mRNA, because the 16S rRNA have a sequence called shine. It has a sequence which will be a complementary bind to the mRNA, and within the mRNA the sequence which complementary able to match with the 16S rRNA is the ribosomal binding site, also called as sine Dalgarno sequence. So 16S rRNA is very important for binding the ribosome to the mRNA, and 23S rRNA is important for the peptidyl transferase reaction, because within the 23S rRNA the peptidyl transferase center is present, which catalyze the formation of peptide bond between the two amino acids in the tRNAs on the A side and in the P side. Okay, so this was all about the prokaryotic ribosomes. That prokaryotic ribosome is made up of 50S and 30S. Okay, the 50S is the large ribosomal subunit and 30S is the small ribosomal subunit. The 50S ribosomal subunit is made up of 23S rRNA and 5S rRNA, including 34 different types of ribosomal proteins. The small ribosomal subunit, that is 30S, is made up of 16S rRNA, and the main component is 16S rRNA, and about 21 different types of proteins, ribosomal proteins, are found. Now let's talk about the ribosomes that are found in the eukaryotes. The ribosome that are found in the eukaryotes, they have also one large ribosomal subunit and one small ribosomal subunit. So suppose this is the large one, this is the small one. The large ribosomal subunit is called as 60S, whereas the small ribosomal subunit is called 40S. And together, this ribosome, together the eukaryotic ribosome is called as 80S. Well, again I am repeating that here S refers to the Swedberg unit, which refers to the rate of sedimentation. Okay, how much the rate of sedimentation during the centrifugation process in the tube, in a centrifugal tube. In this way we determine the size of the particles. Now the large ribosomal subunit, that is 60S, found in the eukaryotes is made up of 28S rRNA. I hope I can write here. Then 5.8S rRNA and 5S rRNA. Okay, so the large ribosomal subunit found in the eukaryotes, that is the 60S, it is composed of 28S rRNA, 5.8S rRNA and 5S rRNA with 40 different types of ribosomal proteins. Now about the small ribosomal subunit in eukaryotes. The small ribosomal subunit in eukaryotes is made up of especially 18S rRNA, including with 30 different types of ribosomal proteins. Okay, 30 different types of ribosomal proteins. I hope you have understood what I have said. So these are the components that are found within the ribosome. Okay. Now, normally these two ribosomal subunit, the large and the small ribosomal subunit, they remain united with each other due to high concentration of the magnesium ions. So here magnesium ions are help to uh, unite the large and the small ribosomal subunit. Now, when the concentration of the magnesium ions reduce in the matrix, both the ribosomal subunits get separated. So, it's due to the concentration of magnesium ions that causes these two subunits to remain joined together. Okay, if the concentration of these magnesium ions reduces, then both the ribosomal subunit get separated. Okay, and if I say in case of prokaryotes such as in bacteria, uh, in bacterial cells, these two ribosomal subunits are found to occur freely in the cytoplasm, and they unite only during the process of protein synthesis. If we carefully look at the prokaryotic, if you prokaryotic cell, there these two subunits, the large and the small ribosome subunits. Of course, the large is called 50s in prokaryotes and small is called 30s. So these two subunits of ribosome freely roam. They freely roam, occur in the cytoplasm, and these two subunits unite only during the process of protein synthesis. At high concentration of magnesium ions in the matrix, the two ribosomes called monosomes become associated with each other and known as dimer. So now if with the in presence of magnesium ions, if these two ribosomal subunits unite, we call them as dimer. And during the protein synthesis, these ribosomes, many ribosomes are engaged, are aggregated due to a common messenger RNA to form polyribosomes or polysomes simply. So polyribosomes look like this. If this is your RNA, okay, many ribosomal subunits unite, okay, and unite, okay. I am writing quite large here. It will be a little bit small, the small ribosomal. So many ribosomal subunits unite to the mRNA and start synthesizing the amino acids. So these red one are the amino acids, the polypeptide chain which is getting synthesized from the um, ribosomal subunit, and it's moving from 5 prime to 3 prime direction during the protein synthesis, and together many ribosomes we are seeing here, they found to associate with the single messenger RNA and start synthesizing the amino acids. Well, you can see the amino terminal end here, which is getting emerged from out from the ribosomes during your translation process, during your protein synthesis process. Okay. So this was about a bit of about the structure of the ribosomes. Let us do more about ribosomes in detail. Okay, more information about ribosomes. So if I say broadly about the structure of ribosomes, ribosomes consist of your number one ribosomal RNA. It also consists of ribosomal proteins, which has not been discussed so much in detail. Sorry, ribosomal proteins. It will be ribosomal proteins and some metallic ions which catalyze the association and dissociation of the ribosomes. Let us start with ribosomal RNA. We have already discussed with the ribosomal RNA. Enough information has been given. Still, I am a little bit repeating. Well, the 70S ribosomes, that is the ribosomes which are found in prokaryotes, have three different types of rRNA. If you recite them, memorize them, which I have said before, what are the different types of rRNA which are found in the prokaryotic ribosome? 23S rRNA, then 16S rRNA, and 5S RNA. These are the main ribosomal RNA that are found within the prokaryotic ribosome. In the large and small subunit together, I have said. The 23S and 5S rRNA, they are found on the large ribosomal subunit in prokaryotes, while the 16S rRNA found on the small ribosomal subunit in prokaryotes. Okay, as I have said. Okay, 23S rRNA. The 23S rRNA, if I say, They consist of about 3,300 ribonucleotides long, whereas the 16S rRNA have about 1,650 ribonucleotides, and 5S rRNA have about 120 ribonucleotides only. But it is made up of ribonucleotides. The rRNA obviously made up of ribonucleotides. Ribosomal RNA is made up of ribonucleotides. Okay, so this was about the prokaryotic ribosomes. Okay, what about the eukaryotic ribosomal RNA? About the eukaryotic ribosomal RNA, if I say the eukaryotic ribosomal RNA has how many types of eukaryotic ribosomal RNA is there? It has 28S rRNA, it has 18S rRNA, it has 5S rRNA, and 5.8S rRNA. So, four different types of rRNA are found within the eukaryotic ribosomes. The 28S and the 28S, the 5S and the 5.8S, these three RNA found on the large ribosomal subunit in your eukaryotes. Okay, while only the 18S are RNA found on the small ribosomal subunit in eukaryotes. So again I repeat, the 28S rRNA, the 5.8S rRNA and 5S rRNA found on the large ribosomal subunit in eukaryotes, whereas the 18S rRNA found on the small ribosomal subunit in eukaryotes. Okay. Okay, now about the if I say mention about the nucleotides, that how many ribonucleotides make up the 28S rRNA. Um, well, if I say about the nucleotides, not so much information has been given. Well, if I say about the 18S rRNA, the 18S rRNA, it consists of 2,100 ribonucleotides. So 2,100 ribonucleotides make up your 18S rRNA. Well, the molecule of 5S rRNA is made up of 120 nucleotides only. So 120 ribonucleotides make up your 5S rRNA, and the other RNAs has not been mentioned. Well, one important thing has been mentioned here, that the 18S rRNA and your 28S rRNA, they have a they have um, they are composed of ribonucleotides. That's good, but it has it within the they have much more about methyl groups. They contain so much of methyl groups present, mostly at 2' position of the ribose sugar there is a methyl group present. So 28S rRNA and your 18S rRNA has methyl group. Okay, it consists of so much of methyl groups present, particularly at the 2' position of the ribose sugar within the ribonucleotide of this 28S and 18S rRNA. Okay. Now the 5S ribosome, if I say the 5, oh sorry, not this one. So now I will talk about another type of ribosomal RNA, which are the ribosome which is found within the mitochondria. Yes, mitochondria also have a ribosome present. They synthesize their own mitochondrial proteins. Okay, but most of the proteins come from the nucleus. Okay, from the translation that is done outside the cytoplasm, in the cytoplasm. The 550, that is, so the mitochondrial ribosome is called as 55s ribosome. So the 55s ribosome, if I write in a good way, it will look like this: 55s ribosome. This ribosome is found within the mitochondria. Now, this mitochondrial ribosome, they don't have this 5s rRNA. They lack 5s rRNA, but they have 21s and 12s rRNA. Okay, so just the opposite of 21 is, so the 55 is ribosomal RNA, the 55 is ribosome found within the mitochondria, and mitochondria have 21s rRNA and 12s rRNA. Okay, the 21s rRNA occurs in large ribosomal subunit, that is 35s, and while 12s rRNA found within the small ribosomal subunit or 25s. So if I say in a simple manner, so the ribosomal RNA, the ribosome which is found in mitochondria is 55s. It is 55s. Now this is divided into two subunits, as we know. The large ribosomal subunit is called 35s, while the small ribosomal subunit is called 25s. The large ribosomal subunit have 21s rRNA as the major rRNA component, while the small ribosomal subunit have 12s rRNA. Okay. Now the sedimentation coefficient of ribosomes, ribosomal subunits, rRNAs, and the number of ribosomal proteins of certain respective organisms has been mentioned here in a table that I will show it to you. Uh, just just in a while. Okay. So let's see now. So this is the structure, these are some characteristic of ribosomes of various organisms. Like you can see, I have already mentioned that prokaryotic ribosomes uh, is normally known as 70s ribosome, made up of large ribosomal subunit as 50s and small ribosomal subunit as 30s. Well, prokaryotic ribosome is composed of 23s rRNA and 5s rRNA. In case of, it is made up in the large ribosomal subunit, while the small ribosomal subunit have 16s rRNA present. About 21 different type of proteins are involved in the small ribosomal subunit in prokaryotic ribosome, and about 32 to 34 different types of proteins are found within the large ribosomal subunit in prokaryotes. I have already mentioned about the eukaryotes, but let us see of other things, like for example in let us say in case of plants, let us say in case of fungi, better to see. Well, in fungi, the small ribosomal subunit is 40s and the large ribosomal subunit is 60s, equivalent to your eukaryotes that we see. The small ribosomal subunit has 18s rRNA present as an important ribosomal RNA, whereas the 60s rRNA, whereas the 60s large ribosomal subunit is made up of 25 to 26 a 25 26 or 26 is rRNA, 5 is rRNA, and 5.8 is rRNA. Okay. Then in case of protozoa and some other protists or protistas, They also have same type of composition of the ribosomal subunits and their ribosomal RNA. Okay, so I hope you have able to understood what I have tried to mention with you about the ribosomal RNA. Now let us discuss about the ribosomal protein in detail. Now we will talk about the ribosomal protein. Each 70S ribosome of E. coli, that is in bacterial cell, well, E. coli is a common organism that we normally we study about, and we represent them in case of prokaryotes. So in 70S ribosome of E. coli, it is composed of about 55 different ribosomal proteins. Out of 55 ribosomal proteins, about 21 different molecules have been isolated by various scientists from the small ribosomal subunit, and some 32 to 34 different proteins are isolated from the 50S ribosomal subunit. So as I have mentioned before that the 70S ribosome is differentiated into large and small ribosomal subunit 50S and 30S. The 50S ribosomal subunit has been used, has been isolated by many scientists and found about 32 to 34 different types of ribosomal proteins. Whereas a small ribosomal subunit, about 21 different proteins, ribosomal proteins are found. Okay. Okay, so it has been observed. Now this actually observed with the help of density gradient centrifugation. In this way we observe the different types of proteins. We actually also, by isolating them, we see their characteristics. Okay, now most of the ribosomal subunits into their component rRNA and protein molecules. Okay, we know the structure of protein molecules and rRNA. This has been mentioned by many scientists. When both of the small and the large ribosomal subunits are dissociated by centrifugation, both of them in a gradient of 5 molar cesium chloride. So we have used 5 molar cesium chloride solution for the density gradient centrifugation, and they have observed many other proteins as well, which shows their relative density, relative sedimentation rate. Okay, after a particular sedimentation time. Okay, which has been observed. Well, not so much information has been given about ribosomal proteins so far. Up to this much has been given. Now, these proteins, if I say about a function, what is the function of what is the function of this different type of ribosomal proteins? They are all evidently play an important role in catalytic activity in your protein synthesis. Okay, so they actually these ribosomal proteins play a central role in catalytic activities of ribosomes in the process of protein synthesis only. Various ribosomal proteins have been found to may enhance the catalytic function of rRNA in ribosomes. So These proteins have only catalytic activity. Okay. Now metallic ions I have already discussed a little bit that with the help of magnesium ions these two ribosomal subunits unite. Other ions which are found is your calcium ions and manganese ions. These two ions are also observed. These two ions are also observed in your ribosomes. Okay. So this was about the structure of ribosomes. We will, in the next video lecture, I will discuss about the function of ribosomes, including their role in protein synthesis. Okay. So that's all for today's lecture. If you have some doubts regarding this lecture, you can ask me. Okay, and for that, what I have mentioned here about the ribosomes, that ribosomes have three sites. I have said now, the A site, P site, and the E site. Will just go through the lectures provided in your junior classes about the translation process, and we will discuss the translation also in your third semester when we will deeply go into the concept of your translation process in eukaryotes, in prokaryotes. Although this has been mentioned, this has been already I have uploaded in my YouTube channel. I will share it to you if you require that video. This will not come in a syllabus, but the role of ribosomes in protein synthesis is very important. Uh, that has to be memorized also. I will give a brief description of these ribosomal functions here, and for the other information you have to go. I will share my translation video lecture where I have point by point I have mentioned that how ribosomes play an important role in protein synthesis. Okay, so I will share it my video here, and also I will try to explain the function of ribosomes in detail in the next class. Okay.